Alrighty, so welcome everybody to today's Emergent session on open workflows. Today, these lovely folks are going to be talking about open community-driven software pipelines and giving us a retrospective look and future outlook. So I would like to introduce Erin, Dickie, Karolina, Finc, Oscar, Esteban, Satrajit, Ghosh, and Tom Nichols. I would like to thank you all for being here and for taking the time. And with that, I will let you take it away.
Great, thank you. Well, thanks everyone for taking time this afternoon, morning, or evening, or whatever time it is where you are. So just as a slight background, I guess if you go back and you look at what used to happen is that in neuroimaging, people basically built their own tools, and each lab had their own custom software. And then in the 90s and the 2000s, a small number of packages came into use that were actually became widely used. So SPM, AFNI, FSL, FreeSurfer, these are some packages that gained wide user bases, but by and large, were not openly developed projects. And so most recently, and I think it's an exciting development, I think it's going to drive a lot of the conversation here, are truly open projects that are trying to take the best available tools from a number of different packages and try to sew them together. And so most prominent of these probably people have heard of as FMPrep or FMRI-QC is just one example. And of course, this is all made possible by the glue that is NiPipe. That is a way of using and interfacing lots of different packages. But then I ask the question, so what if there was some killer tool that was made from that? You know, for example, should someone come in and decide, well, FMRI-Prep, that seems to nail that problem. I think we'll just use that. Everyone stop using what you're using and start using FMRI-Prep. But I don't think that's where we are. So I don't know, who wants to take it from there about what's the long term? How do we, as a community, we're always going to have some projects that are developed by a small number of people who are engaged with the community, but it's not necessarily an open development versus other projects that are open. How do you think these are going to sort of coexist going forward? Let's jump in.
So perhaps the question is why we got where we are right now. Following up with your historical view of how things happened, maybe I can share my experience with FMPrep and why I think it's been quite well adopted by the community. And I think FMPrep found a niche, or maybe it was, at the moment we sensed it was a niche, but in reality it was a bit of a hole in the community because when people start playing around with neuroimaging or in any basically any neuroscience project, there's a lot to learn, and people need to have a good coverage of all the neuroscience background. You really need to endure a PhD in the matter. But also at this point in time, you also need to know a lot about computing and computer vision and processing and all those concepts. And having that in your curriculum is really hard. And basically research programs don't cover for all those areas at the same time because probably it's impossible, or we really need to think about that. So FMPrep basically came to solve that problem. The idea in the beginning was, hey, I just acquired this data. I need to run some standard tool and get it ready for analysis. I just want to dive into analysis. And that was basically the main trigger for FMPrep. And that's why when we talk about workflows, I usually try to be very practical in terms of whether it is solving a basic problem for which you can find somewhat of a balance or a trade-off between the different opinions and options in the world to solve that problem, and then give it easy for the user to apply. And I think NiPipe is a key tool to achieve that because it allowed FMPrep to combine mix and match from FSL, AFNI, FreeSurfer, all the habitual suspects in the field. And I think for FMPrep, that was the main success point. I don't know if you guys agree or whether it is a reasonable thing.
I think also it was helped by that there's less consensus on resting state fMRI. Most people were rolling their own, and so they weren't committed to using FSL, Esteban, AFNI, whatever, because they're going to do it on their own anyway.
Yeah, I think actually it felt like there's more consensus on resting state in terms of there was more, well, compared to now that we're moving into DT, like diffusion weighted, and we're finding like very little ability to figure out where to start even in terms of what the pipeline should look like. It felt like when you looked at fMRI prep from the beginning, there were some very clear steps that everyone could agree on that had been essentially established in the last 10 years. That everyone agreed were like good things to implement. And that actually some of them were really hard to implement, but fMRI prep made them really easy to run without having to learn about some other group's package in order to get that extra processing step that might not have existed in FSL or SPM. Such as some of the surface mapping steps or some of the distortion correction steps.
So I wonder, maybe Satra wants to jump in on this. So let's say next year, the MR people, they figure out how to implement the parabolic 4-dimension, multi-band holographic, acquisition method. And suddenly there's this new type of data. And how are we going to decide what's the best way to sort of analyze that? And how do we keep these things current as things change?
And I think I'll pick up on a couple of things that both Oscar and Erin said in responding to that question, which is it's kind of about encapsulation of knowledge in these open pipelines. It used to be the case that the knowledge was kind of concentrated from what Bob Cox and folks knew, or Steve Smith and MJ knew, or Guillaume and Carl and John knew in terms of how these packages were developed. But these open pipelines provide a way of kind of encapsulating the community knowledge into some of these things and helps move the community forward. And then speaking to your point, Tom, we're always going to be inventing new technologies and new acquisitions. I think that's what science is, kind of trying to probe what we don't understand through the development of new tools. And in some ways, just as those holographic multi-band imaging things are going to be new tools that explore and provide us with insight into some other types of data, the encapsulation of knowledge allows us to process these things in ways. So I think part of the goal of open pipelines should be about how do we process these new data types and how do we process things we know well. And the open pipelines help in both of those contexts. One of the things we have discussed in the context of fMRI prep and NIPIPE and other things is the notion of a system of plugins that allows you to extend kind of core features into more specialized things. And it could be sequences or it could be lifespan. So fMRI prep being expanded to neonatal studies would be very different from the regular bread and butter of studies that fMRI prep is supposed to. We also see that some of the extensions in hardware or imaging provides new challenges for software itself. Things that used to work don't work anymore. And so it opens up, again, a process for development of new algorithms. So now FreeSurfer is able to do processing on 1-month-old babies. It did not used to do it when I first started using FreeSurfer, and maybe not even 5 years back. And so I think the ability to kind of have this knowledge out in the open as pipelines that are easily usable is really crucial for the field to move together. So I'll kind of stop there and say the reason for open pipelines to me is encapsulation of knowledge and for the easy ability of people to redo it. The one last thing I will say is that I always think people should write their own scripts. It's an educational process. But we don't ask people to write the FFT algorithm in their class. But for a math student or a computer science student, they might still go through the exercise of doing so. And that, I think, should still be encouraged. But there is a reason why we use libraries for doing some of these things. And that's where these consolidated pipelines may still be preferred to the educational process of having to go through and write those things?
Yeah, I think it's important, or I think it's something we've learned with the experience of FMPrep, is that FMPrep is a product. It's not you know, a scientific result, or it's just something you can choose to use. And of course, if you are taking, I don't know, a course on photography, and you can maybe learn to do the right thing and then post-process it on your own, or you can just buy a license of Photoshop and improve all your pictures after the fact. So if you want to learn, you definitely don't want to use the Photoshop route. But if you maybe want to have, you know, you need to process a lot of pictures, and that's not the point of what you are learning, then Photoshop might be the right tool for you. So it's always a matter of deciding whether the tool is right for what you want to do.
I think it's always going to be an issue. Karolina?
Yeah, I would also like to add something to what Oscar said. So at one point, learning methods, learning all the available tools can be difficult to newcomers. But there's another level of difficulty when you start to use some tool and then you see that there is like hundreds of thousands of parameters you can set up. And I think what's good in such open pipelines such as FMPrep is that it reduces this complexity, and you just trust this. It has its good, like good and bad sides, but it reduces this flexibility of trying different stuff until you get some results. So I think it's what's, for me, it's in particular like particularly important in FMPrep and such tools as FMPrep.
Well, this, I think, is one of the biggest tensions, and I completely agree with you that when there are areas that I think are relatively settled, I think it's good that we have a reference way, or a small number of ways to analyze something. But I think it's also going to be true that for any one particular situation, any one particular field strength, type of population, type of scanner, there may be a different combination that is better for that. And so this is this tension of it's for people that do not have the resources or the facility to do an exploration, and maybe some ways shouldn't. It's good to have that, but at the same time, what about finding that good match, and what about, again, is this the best, as things evolve, this is the best thing, and should we be looking at different ways of analyzing things? And that's this issue of plurality. If you want to weigh in, it's a can of worms in a way.
Yeah, before we go there, one thing I want to say, which is one thing that I really liked about FMPrep or just BIDS apps in general, was that sometimes the people who developed the tools, they say specifically in their paper, if you have this data, do this, and if you have this data, do that. And most of the end users never read the paper to realize which parameter they're supposed to be fixing, because they don't have enough time to start reading all the physics and the computational science. But one thing that BIDS can do is actually just troll or read the actual data itself and try to figure out which situation and which parameter is most appropriate. Which is something that's nice. You can actually just do that and then inform the user that we picked this because of that reason.
Yes, I definitely believe the BIDS and BIDS apps specifications have helped minimize the need for human intervention, and that only can be good. That's true, and but also coming back to Tom's argument, what I think what you said brings up is we need to really thoroughly test and set some boundaries to where these workflows work well and can be used. So that's the next question, and I'm the first to be honest and say I don't really know which kind of data would be, other than obvious things like babies, would be escaping to FMPrep right now. I do know that if you have, for example, some of these HCP recommended scanners, you probably want to use their HCP pipelines instead of FMPrep because your processing is going to be better. But other than that, and even though we have tried, right now there is a big challenge in front of us to, not only provide these open workflows, but also give with them an idea of where's the domain of inputs those work well and where you shouldn't use them at all. And I think that's going to be the next challenge.
I completely agree. And I think you use the word well, Oscar, and Tom used the word better. And I think as a field, we don't really know how to quantify those two words for many, many things. We don't have a lot of ground truth in a lot of different settings. And so we're kind of looking at it as how well does our results synthesize knowledge rather than when you process this, you should get this exact volume of something.
Yeah, and we also chat about you make this difference between well and better, and maybe the word is sufficient.
Yeah, one thing that and this is sad because I love the H3 pipelines, but I have to admit I don't know if actually HTTP pipelines are more appropriate for data that's using those settings, because HTTP pipelines has a very obscure QA process still that is not necessarily being done properly in all labs compared to FMPrep, which actually shows you a frame for every pre-processing step that could go wrong. And we know the case with ABCD, where had they opened like susceptibility distortion correction pages for every person, they would have caught the errors that were happening in the Phillips scans.
Yeah, that's a really great point. Is that there's I think a successful tool has this transparency or so openness to each of the individual steps, because that's essential. That there's always going to be data that doesn't work well, doesn't fly, and you want to know what that is. Yeah, to me it's a very, very tricky thing, because there's good enough and then there's really good. And I think we definitely need, and Satra and I were just on a different call recently talking about this, we need some reference data sets that we all sort of agree to grind through and sort of agree this is good enough, this is really good, and this is not good enough to get some benchmarks. Because right now we will never have ground truth, but that shouldn't mean that we shouldn't take some data sets and just say, let's see how we do, which ones do better or worse on different metrics. And it's in some ways, these are completely self-evident things to do, and you would do it for any one step. And I'm sure, for example, MR physicists, that's their life is like getting the best acquisition or the best reconstruction. But unfortunately, we as end users actually need to concatenate a whole mess of different choices and steps together, and need to have some practical solutions at the end of the day. I mean, I'm a statistician. I just want the data and start to analyze it. In some ways, I don't want all these questions, but we are responsible for all those intermediary steps.
I think you gave the perfect example, because physicists are, every day coming up with new sequences, but in the end, only a bunch of them are used because, this sequence works better in this scanner rather than this other vendor. And it's exactly the same problem. However, because we need to set some line where we are not going to, question everything, we assume that whatever the scanner spits out, that's what you have. And maybe one of the things we are promoting with FMPrep and iPreps is let's push that interface farther after pre-processing, and let the researchers be creative in the analysis part. It's true, you might not get the best of the data if you assume these standardizations, but what you need, you really need is leverageable data that, if the data keeps the effects you are searching for, then analysis should be able to capture that.
So we just had a comment from Blaise Frederick, who's actually echoing that, which is saying, one of the great things about FMPrep is not necessarily optimal, but it raises the floor. It will probably give you something better and more consistent than picking parameters at random. So that's a good point. Question for Satra and Oscar. how do we know when a standard processing pipeline like FMPrep does capture or do what it's supposed to for a new data type, which I guess circles around some of the questions we had, but it's a sharp question. How would we know?
I think this is where the stochastic process of science has to kind of kick in. We've done that in various ways in different labs about experiments, right? You are exploring some cognitive function. Many people do variants of the experiment, and do you kind of generalize a concept out of it? And I think the same kind of thing has to hold for open pipelines or pipelines in general, which is how do we consolidate that this is a good pipeline for a given data type? So the ideal thing, and I think this is where the ecosystem of things that are coming online is really helpful. So there's BIDS and BIDS apps. There is Neurostars run as an open community platform where as people try these things and come up with different errors, that also provides feedback as to when something works and does not work. So if you were collecting a new data type and you ran FMRI-Prep through it, I would just take it as you've run FMRI-Prep through it. I would not take the output of it as being that's how it should be. Now, if 10 other labs collect that same data type and they run FMRI-Prep through it, now we can start looking at what's consistent and what's inconsistent across these outputs. And that's how we would consolidate knowledge. Fortunately for us, neuroimaging has been around for about 30 years. And so we have consolidated some pieces of knowledge around it, but we still have a long way to go in terms of consolidating it in the context of this chain of complex workflows. I think we've done it at the level of some tools that are pieces of these workflows, but not at the level of the complexity of interactions that comes from your operating systems, your software libraries, your algorithms that do different things, your modeling error assumptions at the statistical level, and then finally, the question that you're trying to answer. The generalization problem, as Tal would put it if you were here.
A good comment from Cyril was just pointing out that optimizing parameters means you have an objective function to work towards. And of course, we don't just have one, we have many, and these are complex data, and you can optimize many different things, you know, the contrast to noise, statistical significance. Yeah, so this is part of the challenge that gets to the original question. Question from comment from Fidel, who says that I don't think it makes sense to even speak about good or better in general. These terms will always be relative to the goal to obtain. Well, so it is a high dimensional thing. There are many different facets, but I think we can say, I would go out on a limb and I think that you could define some things. You will never get one figure of merit that captures everything you want to, but you certainly could make some short lists and say these things are really important. And if you can't stand up on these small ones, well, you know you probably got the wrong pipeline.
Yeah, that's what I was referring to when I said maybe it's not about better or well, it's about sufficient. What is that minimum that really didn't cripple your data, I guess.
Yeah, something that just, if you have a method that falls down in some important ways, then you can just say, okay, that's one we're not going to use. So that's probably about the most sure things you can say. You probably can't say these are the really good ones. You probably can say these are some bad approaches or bad approaches in combination with certain set types of data. I mean, one type of data that I'm actually personally involved with of late is old clinical data. So this is not functional. This is actually structural MRI data. And that's something else that we found that with MRI-Prep, it's okay, but there are some funny quirks that it can be a challenge. So it's just yet one more work case. Randy Golub has a comment. I like the suggestion made by the panel to have a shared benchmark data set. Yes, definitely. Benchmark data set for testing workflows. That is something that is desperately needed. One starting point are the open FMRI data sets, and of course now the NARPS data set. Of course, full disclosure, I'm one of the project leads on that. And it was, I think it's a great initiative to have a common data set that now has been already analyzed by 70 different teams. And then, we can have more people. And then again, we have the heavy lifting to decide what are the metrics to use to actually compare those.
So can I add something about the data? And actually, I had a question for you all about data. So I'll first mention that Open fMRI or Open Neuro gives you a good, in fMRI at least, gives you a good picture of the diversity of data. It doesn't capture all the possible different data types you can find, but it gives you a picture, which we have used in FMPrep to try to stress test it. But it doesn't tell you, if you run it through Open Neuro, it doesn't tell you whether, you know, sorry, I think it's mine. Okay. It doesn't tell you about the results, whether they are acceptable or not. So the question is, how you guys think we need to put together this benchmark data set? How could be that data set realized? What are the requirements? Maybe we just need to focus on a single subject, scan a lot of times with just one scanner, or maybe we want to stick with a vendor, or maybe we want to have a small sample, or we want to do a biobank. What is what would be the route here to move forward?
So Derek, Pilsner has a great comment, which is you know, that the optimization, the invalidation and benchmarking in the pipeline is really important. But in particular, if you do this across heterogeneous data types, you'll then be able to find steps that are hopefully optimal across different parameters. Now, unfortunately, there are probably a lot of cases where different parameters are optimal for different settings. But to the extent that you can find any parameters that generalize, I think is something to look for.
Maybe to answer that question, it's if it's about metrics, it's not just at the pipeline level, right? It's even at the data acquisition level. So we now, a lot of things that FMPrep has addressed is about how it can process things that's been collected over the history, in some sense, of fMRI. But as we move forward as a field, could we consolidate? The HCP effort was one step towards that, right? How can we consolidate some of the acquisition? Does everybody need to use the most niche acquisition possible? Or could we get to a point that a particular acquisition is okay for 80% of the experiments and not the others, where you may need to still push the boundaries? So I think that's one way we can think that open pipelines don't exist in isolation. They kind of exist alongside data and diversity of data. So to the extent that we can, as a community, share more data openly, that allows for these pipelines to get better and for us to evaluate the metrics. I think part of it is I would not be able to say what some of these metrics should be, except within some bounds of some specific use cases or applications.
Well, and there's a lot of discussion in the comments about that, exactly. So what would be ground truth? And some people there are some data sets with task fMRI where you have very strong effects, and that can be a starting point. But it is true, you know, with human data, you don't have great options. Although one person, so Derek followed up that there are reproducible replication data sets, test retest. So HNU1 and HNU2, I'm not familiar with those, are test retest data sets. So that's another thing. And it is still just, you know, consistency, and that's not perfect. That's not everything, but certainly consistency is one measure that you could use as something to optimize.
Yes, I agree. The vibration noise in these pipelines is just something we don't know how big it is. And yeah, right now we're having a collaboration with Mike Milham at Childmind Institute. They're poking a lot of pipelines. FMPrep is one of them, looking at how the results, using this particular data set Derek just mentioned. And they are looking at how repeatable the results are, which is not always what we would like to see.
Well, some of you are probably familiar with the work by Greg Kjar, which I try to forget exists, where he shows that if you simply change some very low-level numerical libraries, your results can change. And we've already known that operating system changes can influence your results. So it's always have to bring that up at least once each year. Let's see, other comments. Other people think that test retest is a good proxy for truth, but again, it is just a proxy. let's see.
Yeah, maybe while you bring up some new questions, I would like to hear from Karolina about, because I remember when she visited us in Stanford in the beginning, she had very strong opinions, and I believe they are very, very well grounded about, you know, the risks of these workflows. And I believe we should devote some time to talking about these risks.
Yes, so I'm working mostly on functional connectivity, and there is a problem with denoising, and that was my idea during my stay at Stanford to build a pipeline which will try different parameters. Like first, I did a research to select like best, like most common pipelines from some methods paper, and then we built a workflow who iterates over all pipelines and then returns some quality measures. And for functional connectivity, is as the most common one is correlation between of frame-wise displacement and functional connectivity. So we did something like this. We're still working on this. So maybe this is kind of example of multiverse analysis. So you run your data, you run your processing, your denoising on multiple pipelines, and then have some quality measures, and you can either run then your analysis on all the noise data or select one which is the best. So I don't know if this is the best solution, probably not, since we don't know ground truth, as you said, but it's sort of trying to handle this problem of selecting best pipeline.
For univariate analyses, there's an idea called the specification curve. It's a really neat idea. If you haven't seen it, you can go Google it and look it up, which basically says for an effective interest in, for example, response time analysis, there are a number of ways you can analyze it, different interactions, different nuisance variables, transformations, what have you. But if you line them all up versus your effective interest, so effect in a you say the mean difference in response times for an intervention, and you get a big curve of all the different results, and on the bottom you have a barcode that shows you all the different analytical choices that you've done. So in a way, that's in a way what we're trying to do is just in neuroimaging, we don't get a single univariate outcome. We have a whole brain or a whole connectome, and hence the challenge of synthesizing that. I think, especially after having been involved in the NARPS exercise, I do think that multiverse analyses are really going to have an important role going forward. The problem is that they really aren't scalable. I mean, it's just not. I can't see a universe where everyone does every possible analysis on that they do. In part because it's computationally excessive and potentially wasteful of electrons. But also because some of them may be just really some combinations of parameters you probably shouldn't do, but of course, if you're spanning a big setting, then you want to consider that. So it's hard. I mean, that's an area of active work. I think it's really going to be important to do more work in the
Yes, but if we have some method paper which compares different pipelines, and there's no like golden rule, but there's some recommendation for different data types, as in the case of denoising, then you can select some best performing ones and then try to run your analysis only on this particular pipelines, not to all possible pipelines, but only on those which are common.
Yeah. Well, I mean, that and I think that's what I think was informal advice, or what I would tell people going back. Don't, if you do every single analysis on your one data set, you won't know what you can trust. But if you, as an exercise, say for the lab, for a given field strength, for a given type of population, take one data set, beat it to death, figure out what actually works, then you fix that going forward. That I think is the idea I think you're getting at. We do have to be careful about doing a multiverse and picking analysis that has the best outcome that you like, of course, and then you're just opening yourself up to vibration effects.
So
Even
I wanted, sorry, go on. Yeah, I mean,
one thing that was true actually in NARPS, which was kind of I liked about it, was that everyone was given a choice to try to pick which pipeline they thought was best. So it actually already constrained the choices down like all of those obvious like corners of the grid, which were silly choices in the beginning, didn't actually end up happening, you'd hope, or if they happened, it was by mistake. All of those 70 teams were picking a pipeline that they thought was relatively appropriate.
Yeah, sorry, I'm just trying to keep up with all the comments here. It sounds like there's a big conversation going on.
well, that's good, no?
Yeah, for the audience, we actually don't directly see all the comments. We're hopefully getting this synthesized by Katie. But basically, just Dare pointing out that test retest is just one optimization objective, and Melanie agrees. And discriminability is another. So that's yet another figure of merit you could use. I'm going to use the pipeline that helps me discriminate best between my two groups. Perhaps the very prediction exercise you're trying to do. Cross-validated log loss is another cross function. Sorry.
I'd say one thing, discriminability, I love it across some settings, but in things like volume versus surface, the better you actually align across people, the less discriminability actually goes down because the matrices start to look more similar as a baseline across everybody.
Right. No, yeah, of course you have to do this thoughtfully, right? Yeah, if you get the structural alignment perfect, there's no gradation left, right? Which just means you need to looking in another domain, right, for the effects. Great. Okay, let's see if there are other questions.
Maybe I would like to bring up the risks again. Sorry about that, and give you an example to try to convey what I was getting at. If you look through the mailing lists of FreeSurfer or FSL or FSPM or all of them, you will find that a good bunch of the issues are people misusing, me included, the tools, basically. So most of the answers are, well, you forgot to set this flag, or you did you set the time in seconds, and it expects the tool expects it to be in milliseconds, and all these things. It happens the same with these workflows, but in this case, the workflows also combine all these tools. So the problem is kind of exponentially multiplied. And that was what I was referring to, Karolina, when you first arrived, because you were saying, hey, you are putting a shotgun in the hands of someone who doesn't know how to use it. So isn't that dangerous? And it is. I also can relate the story when, like last year when Jesse Wright, our philosopher in place, was with us. He spent a lot of time with me thinking about FMPrep. He helped a lot shaping it up. And one of the things he usually mentioned is sometimes tools are too easy to be used. So if the tool is too easy, then people will make mistakes, even if they try not to. So sometimes you will need to insert some friction points to force people to go through certain procedures and make sure the tool they have reflected about this parameter, or this is what this tool doing. So I think this point of adding friction to the tools might be even interesting for all these workflows.
That is the AFNI philosophy, as far as I understand it.
Yeah.
Whenever possible, there are no defaults in AFNI, and it's up to you to just look at it and decide. But I mean, the one thing I think that FMPrep does well, and that is all these diagnostic measures. So it's they're very easy to probe and investigate.
But piggybacking on that comment about friction, I think it's not just AFNI. ANTS follows the exact same philosophy, which is, I'm not going to tell you what parameters to use. You have to decide what it is for your specific problem. And I think that is a fundamental context over here, right? That you have to think about how best a given tool applies to your problem. I see Tom changing his background.
I feel like I'm a late night presenter. So as we can see here, we have a storm of hippocampal volumes that's moving into the northwest. And perhaps this is a figure that Satra sent to help. Is it flipped or is it the right way?
It's fine. It's just amygdala volumes, not hippocampal, but close enough. It shows these volumes from FreeSurfer and FSL. And this was a figure I posted in the threads for the issue, just to point out that even at a single tool level, we don't have consistency. And when we're talking of complex workflows, it goes back to that compounding problem. And so how do we get to even taking individual pieces of tools? And so just to explain to the audience, what this is showing is volumes calculated of the amygdala from the same scan across, in this case, mostly the Abide data set, but a chunk of people were outside of it as well, by FSL 5.10, I believe, and FreeSurfer 6. And if they were doing the same kind of measurement, you would expect all of those points to fall on that red line. That would be identical. But what it turns out is you can see two things. The variance of measurements is slightly different between the two tools. And one tool literally says the measurements are almost twice as large as the other one. For a structural measurement, you would not expect this. So that means we need to do something at a more fundamental level with our tools in trying to better understand how do we reach a standard. So we've been talking a lot about metrics at the level of workflows, but I think there's a lot to still be done at the level of metrics for individual pieces where we should be confident that it's doing the right thing, and we're not quite there yet.
Yeah, I mean, these are two peer-reviewed tools, from the two leading groups in the world, and yeah, they're getting different answers, and yeah, it's yet another. This is some work that actually I'm interested in, and my postdoc Alex Bowring is picking up on for task fMRI, because we did some work comparing AFNI, SPM, and FSL, and found quite surprising differences between them. And now that's the next work we're trying to follow through on. What are the origins of the differences? And this also gets to the idea of a multiverse analysis, because wouldn't it be great if you could figure out, instead of the 1,000 combinations of parameters, actually the 100 parameters that actually make a difference in this. We had a question from Melanie Gantz. What about statistically correcting for all the different reasonable pipelines? A real biological effect should then come out for a majority of the pipelines. And so what about a mixed statistic over different pipelines? We have worked on this for PET. I think that's a great idea. That is something that we tried to do in the NARPS project, where we did a synthesis across. It's a trick because, of course, the data are all the same. So, the most basic standard statistical assumption is independence. Well, not only is the data dependent, it's the same data, just being pushed through different pipelines. So that is a really interesting statistical challenge. And so, yeah, so that's one thing I think needs to be addressed with the multiverse analyses, is that how do you synthesize all these different results out of the different pipelines? And again, like Karolina was saying, you could just pick the best one. I think probably some way of capturing the variability. And there are lots of different ways of doing it. When we discussed this on the NARPS, do we account for the inter pipeline variability? That's something we could have done. Instead, we actually just chose to give a sort of a typical. So what would be the average across all these different pipelines, and what would be kind of be a representative effect? But we could have made all of our inferences more skeptical by the degree of inter-team variability, and that would be like a random effects meta-analysis, and that's something that we could do. Great question. Thanks, Melanie. And actually, you should, Melanie, send either post or send me some links on what you've done with PET. That sounds exciting. All right, I'm going to go back to the GitHub, see if there's any other ideas I can mine, unless any of the other panelists.
So maybe I can ask, Karolina brought up a very nice point on the GitHub issue about how people can contribute to open pipelines. And I think that would be worth kind of discussing. Because as these pipelines become more complex, some people find it a significant barrier to come in and make changes. So I think that would be a good discussion for the panel. How do you encourage people, what kind of skill sets, and where they can contribute to different pieces?
I thought you were going to say, or Oscar, you're going to talk about that?
Yeah, I can take the beginning of this. So in FMPrep, the experience is most of the people don't have the knowledge of Python and details of the workflow at the beginning. So the best way of landing them has been via the documentation. We spent a lot of time trying to document the tool, make sure everything is described somewhere, and people have been really good at finding the holes in our documentation and filling them. And by doing that, they not only are contributing to the tool, a very valuable contribution, which is comparable to code, honestly. They also are learning the tool. So because they have to investigate the tool and understand it in depth to be able to describe it, they finally ended up writing pull requests about actual pipelines and actual problems in the code itself. So I guess documentation is probably one of the most relevant landing points. And then the other thing that has helped is the presence in Neurostars. Having Neurostars as a forum where people can go and they are not embarrassed because they know there are people from all levels of seniority in their careers. They just ask, and that engages in a very nice conversation where we need to look it up, find what's the problem, why it's happening, and then many people who ended up contributing to FMPrep, they did just by posting first in Neurostars. So they didn't even know the GitHub repo or anything like that. They just went to a forum where neuroscientists meet, and then they ask the question. So I think when we are leading a project we want to make open and we want people to contribute, we need to be really sensitive and take every opportunity we have to engage those people and get them on board. And I think FMPrep did very well on that, especially in the beginning.
Yeah, no, I think that's a great comment. We're contributing to documentation, and that for that GitHub is just amazing for making small contributions. But also would like to second the Neurostars. I mean, I think most anyone who works with computers probably uses Stack Exchange every day, and it's basically the Stack Exchange for brain imagers. It's great. It's a great community on there and a great way to search for information. Dan Peterson is asking if anyone has more validation data sets that are heterogeneous with respect to scanner acquisition site. And that's a really good point. I mean, so that verges on to this whole other area of harmonization, but that's a really good point. I don't know. I mean, I know there were some projects that actually had traveling human phantoms, a single subject that got scanned with lots of scanners. Those are rare data sets, but I think there are some of them around.
Right. And we should encourage those people to put them out there or provide a way to access them. I think that's one of the things that would really help move the field forward.
Yeah, definitely. But that's just a good point to underpin this, is that we need heterogeneous data sets to do this validation.
Just piggybacking on the Neurostars point that Oscar is, one of the things that I would recommend to the field to kind of do is, when you evaluate something like FMRI-Prep or any pipeline that you see, ask questions about why something was put in place the way it was. Sometimes that's not in the documentation, and that would actually be helpful, I think, for us to include in various things. The other is, we sometimes take a decision not because we know why it is, but because how we've done it for many, many moons. We've lost the knowledge of why we went there in the first place. And that kind of knowledge would be useful to decouple and kind of put it back to the roots. I always go back to trying to find where the resting state processing parameters for the bandpass filter originally came from. And that's an exercise in digging if anybody wants to go back to. And it's, I think, embedded in a figure caption in some original paper.
Or why the exact hemodynamic response function that we use.
Right. So there are assumptions that we build in that kind of have become institutionalized at some level, and maybe it's good to break them apart at points. And this speaks to the vibration testing and other things that have been brought up by the panel earlier. So keep asking questions as to why we took some of these decisions when we built these pipelines.
Yeah, and adding on this, I would like to mention and self-promote a little bit. That's the idea for also for NIPREPs. By dissecting the problem and trying to find commonalities, for example, between fMRI processing and DMRI processing, we are continuously taking out pieces from FMPrep and putting them in a common project called NIPREPs so that they are also usable in the other project we are running, the DMRI prep, which Erin can describe. So, for example, susceptibility distortions is a standalone project within the NIPREPs umbrella right now. And that helps also people to get engaged because maybe they come from the diffusion world, but they don't know, they feel they don't know about fMRI. If you tell them, can you contribute to fMRI prep? They probably say, well, I'm not super interested in that project. But if the contribution is, can you fix this problem in the susceptibility distortion, which will then feed up into fMRI prep and also DMRI prep, they will probably say, yes, of course, count me in. So that's another strategy we're taking now. We're opening these NIPREPs. You can go to the website www.nipreps.org. And you can see all the map of tools that are underneath fMRI prep. And probably many of those will work for you, for your open workflows. So they are reusable and they are thought to be like that. Great. And on a corner, we have NIPipe and also NIFloats. So maybe Satra can talk more about those.
We're going to have to wrap it up soon, though.
So just a quick point, there are a lot of community tools. So ask questions before starting to create something. There's a lot of projects that you can contribute to at different levels. And I think if we build this as an ecosystem, everything gets better. The one thing I'd like to kind of touch on is the NARPS paper a little bit, which is it's not just the multiverse testing. A lot of that is technologically feasible today to automate. And so those are some of the things that we should be moving towards, where existing tools and technologies can be integrated together to provide that kind of multiverse analysis for anyone out there. And if we do that as a community, that'll just help us move forward.
Yes, yeah. BIDS has shown how much the profits of sort of getting things in a machine readable format that everyone agrees on. Yeah. One last comment. There was a comment from the group that the open source traveling phantom neuroimaging datasets are available. They're part of the BURN project, and you can find them on Nitric. So that data is out there.
And Open Neuro.
And Open Neuro. Good.
All righty. And with that, this concludes our session. Thank you all so much for being here. Thank you all so much for listening. And I'm glad we had such a nice conversation about open workflows.